Welcome to Open Mind Integration. Christopher Gunlock here. This is the Psychedelic Harm Reduction class, Part 5, Neurobiology and Psychedelics. And always be sure to check out all 12 parts of the Psychedelic Harm Reduction class available on openmindintegration.com. The usual disclaimer. So this is a really rapidly expanding area of research. The brain is really just infinitely complex, and it takes a lot of time and effort in order to study even just small parts of it. But my hope here is just to provide a few different evidence-based perspectives on how psychedelics affect the brain, just to start to give you an idea of what direction the brain is being taken under the assistance of psychedelics. And we'll begin by looking at some mechanistic effects in the brain, what exactly physically is happening with psychedelics involved. So the first mechanism we'll cover is the BDNF protein, brain-derived neurotrophic factor. It's a hormone made in the body that sends a signal to induce neuroplasticity, or at least the possibility of making new neuronal connections. And they've mapped it to certain contexts, certain behaviors like learning, stress, memory, and indeed psychedelics. So it just goes to show there is a common denominator here. And I think something like learning is maybe obvious, but stress, that's one that we maybe take for granted sometimes. That acutely stressful events, situations that challenge us to a great degree, can be highly constructive and have a big impact on the way we perceive things and our sense of identity and in general just our habits or how we live our lives, much in the way that psychedelics can do that as well. And memory, of course, it's involved very much in any process where you're imprinting new parts of yourself, drawing upon the past, learning from the past. So BDNF is very important. It's no surprise that we see a connection in this study. They basically administered a psychedelic, in this case DOI, as well as the BDNF protein itself, to see what the different effects would be. And in terms of the number of synaptic crossings in the brain, they found that very similar actions with both the psychedelic on its own and the BDNF protein on its own. And then even so, when they combine the two, they'd expect maybe to see an augmented effect, but it was actually quite similar. And that would demonstrate that they're kind of doing the same thing. They're following the same processes, and indeed, they found that the protein synthesis pathways were activated with both just straight BDNF and psychedelics. So, really outlining a potential pathway of action. Another feature they noted was the long-lasting upregulation of the effects, their neuroplasticity, for example. And my guess is that speaks to the afterglow period, right? While the main effects of the psychedelics have worn off, like a day later or weeks later, there is something that persists. There's a little bit more flexibility in your perspective and the way you do things. And here is a little bit of physical evidence of that. So here we have the neuronal synapse, something that's happening about 1,000,000 times per second in our brains. Every little branch, every little synapse is communicating through chemicals and electrical signals. So we can imagine if we were to take psychedelics and the compounds get into the bloodstream, eventually pass the blood-brain barrier, and then are free to then attach to a neuronal receptor. There you can see in green the way the compounds can float around in the gap and then get picked up, and then there's a signal that gets sent through the neuron to then tell it what to do from there, how to interact with other neurons in the brain. But also similarly, we can actually create our own psychedelic-like compounds, endogenous tryptamines here in this case. Tryptamines are the class of compounds that most psychedelics are in, and a lot of things that we can make in our own body, like serotonin, for example. And yeah, those are synthesized in the brain and then released in a similar way so that they can be picked up by a receptor. I also am showing in red there the monoamine oxidase inhibitors that you find in concoctions like ayahuasca. And here showing the protective measure that it can take to prevent it from being broken down and removed from that gap there, making it more likely to get picked up by a receptor. And that's what potentiates the psychedelic effects of something like ayahuasca. Neuritogenesis and synaptogenesis, just fancy words to describe the way our neurons can grow more branches and synapses to make those chemical connections. And psychedelics definitely involved here. So this study is really interesting because they actually took some neurons out of a brain, just had them in a petri dish, administered the psychedelics directly to their neurons, and found, yes, indeed, a mechanistic effect, purely mechanistic, in fact. So you can see very visually there, the VEH is your control group, no psychedelics, and then DOI, DMT, LSD being your psychedelic groups. Showing, indeed, we're seeing some spikes, some synapses popping off, especially LSD there, really kind of showing off for the psychedelics. And yeah, and this is explains a lot as far as what we understand the experience of psychedelics to be like. You know, you get your novel thought patterns, your new ways of perceiving things and understanding and drawing upon memories and associations. These are all just extra brain connections, right? Just things communicating that weren't communicating before. And psychedelics simply make that more of a possibility. And due to it being a mechanistic effect, I think it's relevant to say that there's more going on here in terms of, especially if you're looking to use psychedelics to treat certain illnesses or to accomplish a certain task. It's more than just taking it. You have to direct thought in a direction. Because just having extra synapses and extra spikes doesn't necessarily mean you'll make those connections. The connections themselves, that depends much more on what you're doing, what you're thinking. But also from a clinical perspective, this is extremely relevant. We know that mental illnesses like PTSD, depression, addiction, OCD are the result of not enough connections. You know, there's even findings that the neurons themselves can get inflamed and retract their branches, retract the possibility for more connection, which, you know, a lot of us have experienced that rut of thinking where it's just really hard to just get out of the same pathway of thinking. So psychedelics, no wonder, are helping out with this big time. So on the topic of neuroplasticity, let's bring MDMA into the discussion. Here we see that they're comparing MDMA to the control group with also testing a learning activity. And so they're showing a couple different groups here, the learning activity and without the learning activity for both MDMA and placebo. And interesting findings, we see that the neuroplasticity is actually lower when MDMA is taken, but there's no learning activity. But then a significant jump when we do go into the learning activity. So seeing a neuroplasticity being dependent on whether, you know, what you're doing there, which further demonstrates that, you know, psychedelics, they're creating the opportunity to form new connections, but not necessarily guaranteeing that on their own, as just a purely mechanistic effect usually goes. So why there was actually less neuroplasticity with MDMA compared to placebo, that's very interesting. I'm really not sure, but certainly that the MDMA is not going to be so much like the other psychedelics. It's not even acting on the serotonin receptors in the same way. So likely some differences there. But the BDNF hormone nonetheless was expressed more with MDMA, regardless of whether or not there was a learning activity. So what are these receptors that psychedelics are acting on to cause the effects that we're familiar with? Well, the 5-HT2A serotonin receptor is a big one. You know, things like psychedelics or neurotransmitters will act on, you know, different kinds of receptors, but there's certain ones that do tend to characterize or be responsible for the characteristics of that compound. So in this case, we know that this receptor is responsible for formation of individuality, huge part of egoic constructs, spatial cognition, learning, memory, social interaction. And we also know that this receptor is in the highest density in regions like the default mode network, these executive control networks, the hub networks, that they do that shift from focusing everything internally, you know, filtered through these hubs, or if turned down in the other direction, opening up the possibility for less of those constraints. So it's like that reducing valve features. And so it makes a lot of sense. This is where psychedelics are funneling into, and the effects are very much processed. The way they did this, the way they figured out that this receptor is the one, is that you can actually administer receptor blockers. And so if you know you're blocking that particular receptor, but you give the psychedelic anyway, if you don't get the effects, then you know that's the one that is responsible for the main effects of the psychedelics. So let's take a closer look at what these neural hub networks are and how psychedelics are changing what they're doing in our brains. So to start out, neural hub networks you can visualize as kind of an in-between of your raw data, the things that your brain, your consciousness is generating from either things outside of you, exogenous stimuli, you know, what you're sensing, or things inside you, what your memories, what your emotions are causing you to think and feel. So all of that consider as like this raw data, you know, there's so much coming up, but ultimately what we become conscious of and what we express is determined by these hub networks. What does the default mode network say about, you know, how this feeling, this thought affects our sense of identity, how other people see us, or the executive control network, which determines, you know, what's the best decision for me in the long term. You know, how can I how can I act in a way or express myself in a way that will preserve my sense of realness? So the hub network is that in between of governing what ultimately makes it to the surface. So the default mode network is one that's talked about a lot with psychedelic research, and for good reason too. Here we can see it's not just one part of the brain. It's a collective effort amongst different regions, and you'll see this a lot in general with neurosciences. It's, you know, the brain works in concert together. It's like these groups of regions creating an effect, and extremely complex different ways of entanglement. But the default mode network in particular, you can think of as this constant reversion to self-justification. This process of thinking that we're all extremely familiar with, who maybe just don't consider what it is while we're doing it. But anytime you're just thinking about who you are, what you're doing, how other people think of you, thinking about what happened before, what could happen, a lot of anticipating, a lot of analyzing. Ultimately, this kind of self-preservative way of thinking. And as you might guess, it really kicks in heavy when you're not really doing anything else. It's called the default mode for a good reason. It's just what you default to when you're not doing anything else. And in that way, it has this very strong anti-correlative effect, which we'll talk about later, with other thoughts and thought processes of thinking. And then if you look at, you know, how often we spend in this kind of inactive state where we're just go on autopilot, it's quite a lot. It could be 30 to 50% here. So big part of our functioning. So it's important to look at it. So as one could imagine, then the effect of psychedelics on this particular brain network that governs our thinking of ourselves, our sense of identity, is going to be affected quite a bit. So yeah, there's been a lot of research around this. I'll be kind of dipping into a little bit of it here and to try to provide just kind of basic framework of what's happening. So one way to understand the function of the default mode network is to look at how it might be communicating with other networks of the brain. Here we're looking at the attention and sensory networks. So this is what your five senses are picking up. This is what you're externally thinking about. And so the default mode, this is kind of the egoic senses. This is your fears and insecurities, your desires, your sense of personality. Um, in the situation where you observe or you experience something that's traumatic, the default mode is going to construct a pattern response to it. It's going to determine that maybe it's something you want to stay away from. It's something that the nervous system reacts to in a specific way, and so when that kind of thing comes up again, your sensory networks are picking up on something. The default mode may come in and override what it actually means. The doorbell's ringing, but oh no, you're thinking about, bullets firing and helicopters whirling, and that's an example of how much in control these hub networks can be, and how that can form our entire sense of reality. And we've all had those days, right, when we'd really like to be able to do something productive or feel just kind of motivated. But our brain just can't get out of those thought processes, the overthinking, dwelling on the negativity, stressing but not doing anything about it. This is depression, right? This is anxiety, the chronic mental illness states. And it shows it from the research that it stems from this hyperconnectivity within the default mode network, these hub networks, where if that's all that's active, it's just doing a lot to limit what things we can think about and the ways we can think. It's that rumination, that low self-esteem, and ultimately manifesting in these ruts that just kind of dominate our thinking processes. So attention more generally has a really strong relationship with the default mode network, but it's an inverse correlation or anti-correlation. In other words, when one is very active, the other one cannot be. They're mutually exclusive to some extent. And so here we're illustrating what the brain is like when there's a lot of attention engaged in your conscious awareness, where you're having to focus on something external from you. Maybe your motor skills are activated, you're doing something with your hands, or there's some active problem solving. Anytime you're having to observe things outside of you, creative activities, making art, playing music. I think many of us can attest to when we're doing those activities, we're probably not, ruminating about, our spouse getting in a fight with us, and we're probably not, considering why it is that we're always so late to work, and reflecting on all these, very introspective type things. That kind of thought is just kind of disabled when you're externally having to focus your attention. So then there's the other side of things when the default mode network is more prominent. This is going to limit our ability to pay attention to something outside of us, to pick up what's happening, maybe right in front of us even. A classic example is, you walk into a room with a bunch of people, you shake somebody's hand, and they introduce themselves to you, and you immediately forget their name. You were not paying attention. You were thinking about a million other things, all the other people in the room. Your ego is going to act up and start analyzing and reflecting, and, am I doing the right thing? Is my hair okay? and what you're picking up from your five senses is just getting filtered out. It's been overridden by the default mode and these hub networks. So this is that classic daydreaming look, zoning out, that automatic thinking that a lot of people with chronic anxiety, chronic depression experience uncontrollably even, and just goes to show that if we allow these hub networks to become too prominent and stay in them too long, it may be hard to balance the scale out in the other direction. It may be hard to pay attention in general. We see a lot of that concurrent addictive kind of behavior along with depression and anxiety. So, yeah. So we'll move into the brain imaging research, and I just want to caveat this section with, the pictures of the brains, with the colors showing different regions lighting up. We really need to take these with a grain of salt. We're only able to measure, a couple things at a time, the relationship between them. And so just because the brain is lit up in one graph, it doesn't mean that's lit up in every situation, or that's what every part of the brain is doing. It's usually a very specific relationship that you're looking at. So just try not to overgeneralize too much. So to start, it's all about blood flow. When we're looking at these colored regions of the brain, what we're looking at is areas that are either getting less or more blood flow. And the reason we do that is because that's associated with neuronal activity, the firing of the neurons, the chemical signals being sent to change the way the brain responds. And for example, here we're looking at these blue and green colors being associated with decreased blood flow while on psilocybin, and so showing that there are in fact some regions of the brain that are not as active, and this further adds to that evidence that these hub networks are affected in kind of a down-regulated way with psychedelics, and that's our analytical thinking, the kind of rational processing, referring to ourselves, the sense of identity. And another brain region that was measured here, the parahippocampal network, key feature of the default mode network. Again, decreased blood flow on psychedelics. So these are the parts of our brain that are processing, they're filtering, they're constraining. And I brought in this term, the reducing valve, coined by Aldous Huxley. I think even though it was decades before any of this brain imaging technology, he really, described it in a way that made a lot of sense. That these valves in our brain, these hub networks that constrain what gets expressed into the conscious thinking. Psychedelics have a pretty strong effect on them, it seems. So there's less of that action of filtering and constraining. So global connectivity, this is kind of the other side of things. Again, showing that these brain imaging scans, the colors can represent different things. In this case, it's the areas that are having more connections. Specifically, though, with the seed region, and that's what you'll be seeing in purple there. Notice that the purple regions, they're in the visual cortex in this case. They're the same, the placebo and the LSD, and that's not because nothing's happening there, but it's the seed region that is connecting with other parts of the brain that we're actually measuring, and that's what that kind of yellow, orange colored areas are. That's what's communicating with the visual cortex, and so that's all we're looking at in terms of this measurement. And there's LSD will really lighten things up. So I think this speaks well to the subjective experience of being on LSD. From the visual cortex, there's a lot happening that isn't normally happening on LSD. What you're seeing is more than just what's there, right? You're seeing things change in front of your eyes depending on what you're thinking about. What other parts of your brain are lighting up. That's where kind of that sort of hallucinatory state it gets popularized in the realm of LSD. But psychedelics in general, yeah, your visual spectrum is more than just what's there in front of you, but it's your interaction with it. And this study looking at MDMA, neuroplasticity and fear extinction. So fear extinction is that process of no longer having the same response to a fearful stimulus. In this case, the rats are freezing up when there's something that they're afraid of, and the MDMA being shown to have an effect on that. There's more fear extinction. They're kind of getting over that fear response. So this being very, very relevant to the realm of PTSD, where the brain is still responding in the fearful way, even though there's not necessarily a fearful stimulus there. So this pairs well with the current clinical evidence with MDMA and PTSD. Does a great job. Yeah, it does a great job helping people let go of those fearful responses. They also actually looked at BDNF expression, again, that hormone associated with neuroplasticity and change and learning, and seeing that MDMA does increase BDNF levels, but interestingly, mainly just the amygdala that they're measuring. Of course, again, take it with a grain of salt. These studies can only really say something about that which they measure and not so much what they don't measure. But nonetheless, the amygdala being a big part of this definitely makes sense. So afterglow, that effect after the experience of being on psychedelics is finished, usually like the next day. What's happening in the brain? Well, these folks measured a lot of similarities, actually. The before and after, still a lot of the same parts of the brain being lit up, a lot of that connectivity outside of just those hub networks. But they did also measure, there was some differences, certainly. Areas of decreased blood flow, looking at the amygdala, among a few others. Again, we're looking at those hub networks. They're not as active because they're the ones that are constraining the rest of the activity that we see happening while on psychedelics, and then, of course, after psychedelics as well. But I threw this one in there. It's showing a bit of contrary evidence, actually, that there's some increased blood flow in areas associated with depression. And that's just going to show that the way you're doing psychedelics can mean totally opposite results. Some people, frankly, they get worse after a psychedelic experience. For a number of reasons I describe in my other classes. The way we integrate our experiences can very dramatically change, how open we are. One of the terms I bring up in later classes is psychedelic whiplash, when the ego snaps back really hard after kind of a traumatic psychedelic experience, puts up more defenses. That's ideally what we don't want to see. We want to see folks going through an experience where they're able to accept and surrender to the changes and new insights, so that the ego doesn't feel so offended that it needs to come back with a vengeance and defend itself. But nonetheless, we see that, and I think this study illustrates some of that possibility. So this study was just looking at when people take psilocybin and they describe when they're having a mystical experience, what do we see happening in the brain? And what they found was the mystical experience reported effects were correlated with lower activity in regions like the amygdala and the default mode network. So again, these brain areas responsible for kind of overriding things, for filtering things out, for constraining what ultimately comes to the conscious awareness, well, they're turned down, and as a result, we do tend to see more of a mystical like possibilities. And so that term anti-correlation comes back into mind then here, instead of the hub networks and attention having the anti-correlation effect. Potentially mysticism being something that can be more active when the hub networks are less active. And then potentially the other way, we can imagine when the hub networks are more active, when we're undergoing our more default states of perception, there's more filtering, there's more constraints, then it does make sense that these mystical states seen beyond the boundaries, seen the connections beneath the surface, that being less of your conscious awareness, and I think this kind of back and forth, this anti-correlation, illustrates a lot of the human experience, especially when you're throwing psychedelics in there, that while on the psychedelics, you're kind of forcing these hub networks to quiet down. Yeah, mystical experiences are far more likely. But then when the psychedelics wear off, yeah, well, those hub networks come back into play, and it's, this is sort of mystical amnesia that takes place. So now let's pivot to the topic of endogenous psychedelics, psychedelics that are made in the body. So to maybe some folks surprised, this has actually been researched for quite a while, a number of decades, way back in the 1930s and 40s when, Western countries first started to investigate psychedelics as any form of research. But a lot has developed over time, and I'll get into that here. So we'll start early on. A lot of the research, particularly with DMT, was already seeing a potential connection between measured states of conscious or observed states of consciousness like psychosis, schizophrenia, potentially to have some connection to the DMT psychedelic. And from one perspective, this makes sense because these states of consciousness, these kind of pathologized illnesses, have some similarities to what some people experience with psychedelics. Schizophrenia, perceiving things, outside of your normal senses and your thoughts going to more abstract realms. You'd think maybe there would be a connection, but actually the research for a long time actually had a hard time connecting levels of DMT in the body with symptoms of schizophrenia, for example. Moving forward, they continued looking at the possibility of DMT being in the body, found that in animal studies here in rats. They did find DMT in the central nervous system, and even found that stress seemed to have a correlation with DMT levels, as we'll talk about in a little bit. There's a little bit more evidence to that. So even further, they were able to identify a potential pathway for creating DMT from things that are already in the body. Tryptophan, tryptamine, very common precursors to other things. Tryptophan's in a lot of food. You're going to get it in one form or another. And seeing that in the mammalian cases, that there's a pathway there. You could synthesize DMT using things that are normally just in the body. So we'll talk more about this too. So I offer here Barker's review in 2012. I do think it's a flawed paper. It's missing some things, but as a meta study, I think it does bring together some valid points. One thing really kind of concluding based on the decades of research trying to associate levels of DMT in the body with illnesses like schizophrenia, still not a lot of strong evidence there. And my suspicions is schizophrenia is an extremely complex situation. There's even if even if DMT has some connection to these states of psychosis, it's just one potential thing among many. The brain is full of chemicals and neurotransmitters that are very likely to be involved. But also, I think the evolution of schizophrenia, when you're dealing with it for a long period of time, it changes. It changes your life. It changes what happens to you, which then is going to change the state of your neurobiology. So, Barker really kind of confirming that. We're having a hard time connecting endogenous DMT levels with schizophrenia. They even tried to block the symptoms of schizophrenia by administering a drug that would block DMT synthesis. And yeah, no connection again there. Really kind of a hard way to go about that, but yeah, yeah. Nonetheless, though, we do have a model for DMT potentially being made in the body, and moreover, we can see that serotonin, melatonin, things that we know very well to have a significant role in the human experience and in the brain. Very similar stuff going on to make DMT, potentially the same precursors and just a different set of enzymes to bring about the synthesis. So there's even some papers written where scientists are kind of checking the boxes of what it takes to count something as a neurotransmitter, and DMT really checking the boxes. Yeah, it really seems to have a lot of these qualities that are needed to even consider the possibility of DMT playing a central role in our cognition and the rest of our body systems. So moving ahead a little bit, some more rat studies. This particular lab we'll talk more about, but already beginning to find that there are a lot of biosynthesis of these tryptamines, DMT being one of these main ones. And if you go back and watch part 2 of the psychedelic harm reduction class, show there's a commonality of the DMT molecule with many other things, some of them made in the body. So yeah. Also the complexity again, hard to find DMT in consistent levels amongst different samples. They were looking at, I think, blood, urine, spinal cord fluid, tissue samples, and yeah, it's tough to detect it consistently. Potentially a lot of variables affecting where DMT is in the body, but also just the fact that DMT breaks down so quickly. There's a lot in your body that is not allowing DMT just to like hang out in the blood or in fluids. So the Borgesian lab, a lot of good recent stuff out of here to explain, the possibility of DMT being made in the body. One key paper here, John Dean of that same lab, doing some tests on rats, hooking them up to microdialysis brain probes. I actually got to talk to the guy about how they did this, and really quite invasive. They had to stick tubes in the brains of these rats to consistently run the fluids through a machine that would detect the DMT. And yeah, you can imagine trying to do this on a person would just be terrible. I mean, they killed the rats at the end of the study, unfortunately. And but anyway, they did find some good evidence looking at different tissues of the brain and trying to see are the building blocks for synthesizing DMT present. In particular, they're looking at the transcriptors that make the enzyme that allow the brain to make DMT out of the basic compounds like tryptophan. And they did find that. They found in a number of areas. Notably, the pineal gland was not the only area, as we'll say in a moment. But another interesting point is after they killed the rats, they induced cardiac arrest. They stopped their hearts. And these poor dead rats, though, were actually still creating DMT for at least 30 minutes after they died. So that, I mean, that just gets my imagination going quite a bit. Thinking about the afterlife. These near-death experiences being very similar to psychedelic experiences, and life flashing before your eyes, seeing visions. As you go on to the next life, and the idea that DMT could somehow be involved in that, yeah, kind of makes sense to me. But some other points that came out of this study that I think is good to get across. There was some talk from previous studies that the lungs were where you create DMT, and that maybe like breath work, doing intense breath work, could have something to do with the states of consciousness that you can reach through that method. But unfortunately, this study shows that the lungs actually only have one of the two necessary building blocks, the enzymes that can create DMT from tryptophan. Now, I did get into this with the author of this paper. We were doing an event together, and he confirmed that, while this definitely demonstrates that you couldn't synthesize DMT from tryptophan in the lungs, if there were tryptamine there for some reason, which would take some more research to validate, then maybe, yeah, you could create some DMT in the lungs. So don't be too conclusive on this, but at the very least, we're seeing it probably more likely that DMT is created in the brain tissues as opposed to the lungs or any of these, what they call peripheral tissues. So basically anything other than the brain, they found both of these key building blocks present, so making it very likely that DMT could be synthesized. And then also, as I mentioned, the pineal gland, they actually tested rats that they removed the pineal gland and found that the DMT levels, the overall DMT levels that they were creating was actually about the same. So DMT created not just in the pineal gland for sure, at least in rats, but many parts of the brain. So I offer this paper as a way to provide some potential counterpoints to this idea that DMT could be made in the brain. Nichols, I think, points out correctly that the doses that it takes to have the kind of classical DMT experience, like the drug experience, is hundreds of times larger than what they've measured so far in terms of the doses that the body can actually make, what's made in the brain all by itself. But the data on this is tricky though too. Again, just the nature of measuring DMT is very difficult. It's metabolized very quickly. It's kind of gone, as soon as it shows up. But also that, trying to measure the collective DMT levels in the whole body, in the whole brain, very difficult. So whether or not the doses within the brain are in fact that much lower than what you would see when you actually take DMT as a drug, I think still open to investigation there. But nonetheless, Nichols does point out that if you don't have enough DMT crossing the blood-brain barrier, acting on the serotonin receptors, you're not going to even activate that special receptor, the 5-HT2A. So that there's definitely a threshold you need. If it is going to be endogenous DMT, you need to create enough of it to have a potentially psychoactive effect to change your consciousness. So he concludes that maybe it's like obviously we're measuring DMT in the body, but maybe these smaller levels of DMT serve a different purpose, if not that to have that intense psychological change. And they did find, that the beta receptors DMT acts on as well, and those receptors associated with immunoprotective effects, regeneration, tissue protection. But nonetheless, Nichols acknowledges still there's kind of a qualitative similarity with states of consciousness like dreams, near-death experiences, out-of-body experiences, but that, just looking at DMT, as magical as it is, there's a lot going on. Dopamine, serotonin, they'll have a strong effect on our states of consciousness. So we should also keep that in mind. Also important to understand, as I've been reiterating here, that it's hard to measure this stuff. This research has a long way to go. The ways we're detecting DMT is actually pretty imprecise, and just the nature of DMT being so hard to measure, it really is quite a challenge to be sure about any of this stuff. And there you can see some of the numbers, a detection rate of much less than 1% in urine. Hard to measure. Also in correlation with different states of consciousness. And that's really kind of where this discussion goes, is like, okay, well, in just a regular waking state of consciousness, we probably wouldn't expect there to be a whole lot of DMT. But what if we measure DMT while we're meditating, or while we're doing breath work, or while we're having some sort of, grand mystical experience, and from, any number of methods. Well, as I've described, the methods of measuring DMT are quite invasive. If it's not sticking a tube in your brain, as we'll see, even non-physically invasive methods, it'd be hard to, test in different types of conditions. So the one new area of research in detecting DMT is using magnetic resonance imaging, MRIs, instead of measuring in the physical fluids, using basically energy waves of light to bounce off of the individual molecules themselves, because you can kind of detect very, very specific frequency changes that correspond to specific molecules. So this method has some promise. We'll have to wait and see, what comes out of this. But in order to do this, though, as I said, it's still kind of invasive. An MRI machine, if you've ever been in one, it's actually you can feel it. It's not a passive experience entirely. And so if we wanted to compare, what DMT is doing in the body in, let's say, an altered state of consciousness, again, whether it's meditation, breath work, some form of something to alter your subjective experience, are we going to see DMT levels change? Well, you'd have to be in an MRI machine during those moments to know for sure. So yeah, those MRI machines, it just it really rattles you. You can physically feel the jolt of energy passing through you. And and at that point it's like the act of measuring itself is potentially changing the result of it. So it really just goes to show this is not going to be easy to verify, and that we may be left with just kind of investigating DMT from a more subjective perspective. But you never know. Technology is always changing, and there's always, of course, a desire to enhance our perspective from this methodology. But again, there's a lot of reason to wonder. Near-death experiences, out-of-body experiences, dreams. Is there a connection with DMT in the body? You can't help but wonder. You really can't. But part of this, Rick Strassman was really raving about this in the beginning of the research days with DMT. But yeah, still, the evidence is there, whether or not speculation has panned out in light of new evidence. So long way to go on this. But here I'm just kind of bringing it all back around here with the entropy spectrum. You can see we've looked at a number of areas in neurobiology, the brain regions. We see this high entropy state where psychedelics and these altered state of consciousness, neuroplasticity, that's more on that end of the spectrum, right? And we in fact see certain brain areas respond to that more. We see a lot more of the basic instinct, that raw data coming from our senses, that's unleashed, right, in that high entropy state. And then the opposite, low entropy states, the regions that are more active are these constraining hub networks, a default mode network. It's that self-reference, that just kind of thinking about us as an individual as opposed to the bigger world out there, the bigger universe. So Yeah, and then information feed, again, back to the psychedelic psychology. There's more explanation from that perspective that, these high entropy states, these regions of our brain that are generating the raw data, the raw experience, emotions, and senses, it's feeding into these hub network regions that are processing, analyzing, and filtering. But then also there's this act of suppression, right? It doesn't allow the raw data to come to the surface. So it's really kind of an orchestrated effect, right? Our brain is handling these different states of consciousness in a very intentional way. But when you throw psychedelics into the mix, you can see that all of a sudden you have a lot more opportunities of ways to experience your brain. So good work. This has been part 5, Neurobiology and Psychedelics. Hope you enjoyed. If you'd like to go deeper, openmindintegration.com. You'll find some services to help you along your path. And there's a ton more awesome content in the Psychedelic Harm Reduction course, 12 classes altogether, each one designed to offer some really in-depth practical education on how to reduce your risks, maximize your benefits. And you can view all the slides for every class for free at openmindintegration.com. And there's a bunch of other educational content up there as well. And my personal services, of course, for people going through preparation and integration processes. Here to help folks go a little deeper, be a little safer about it. So please let me know. Thank you.